In this session, we're going to cover the epithelia, so the epithelial cell lining, and its variety of functions. So first of all, we really need to look at an overview of the characteristics and the functions of your epithelial cell layers. Now, your epithelium is the thin tissue layer of cells that covers all of your body surfaces, and that includes your GI tract. Remembering, of course, that your small intestine epithelium is primarily absorbed in the absorption of the products of digestion. But epithelium, as a general rule, also protects itself and us from noxious intestinal contents by the secretion of a surface coating of mucus, for example. So in other words, it's functioning to aid in absorption of what we want to get into the body, secretion, what we're secreting out, physical protection and containment. So it has all those multiple functions. And the epithelia are derived from three different germ layers, which you'll come to later when you start to look at embryology. And if we look at the first outer layer there, you can see the ectoderm, and as it says, forms the exoskeleton. And when we're thinking about that, we're thinking of things like the epidermis of your skin and related glands, lining of your mouth, the nasal and the rectum. And the ectoderm also goes on to form our nervous system. You've got the mesoderm. This is your epithelial linings of the cardiovascular system. For example, your endothelium. So your endothelial cells line your blood vessels, and they have very important functions. And we'll go through that later when we come to the cardiovascular system. But it also lines your kidneys, your gonad linings as well. And it goes on to form your muscular system and skeleton, your cartilage, and of course your blood. And if you're looking at the endoderm, that's the epithelia of your gut, respiratory system, urinary bladder, liver, gallbladder, pancreas, so parts of your GI tract. So when we're really thinking about it, it's also forming the glands, for example, of your digestive glands, your endocrine glands, your adrenal cortex. And like I said, it's your gut apart from the mouth and the rectal or anal canals. So how do we actually classify epithelia? Because they can be classified by many different ways and by their shapes. So when we're basing it on the shape, we can look at these three main types in front of you. So you've got squamous, which if you look at it, it's quite obvious, is flattened. You have cuboidal, which, as is fairly obvious, is cuboidal in shape. And then you have columnar, which is tall and elongated with a nuclei near the basal membrane. And we'll talk about the different types of membrane later on. An important thing I want you to just look at there is you've got this basement membrane. Now, your basement membranes are really just thin layers of extracellular matrix made up of connective tissue with proteoglycans, so proteins with carbohydrates attached, that go on to form a supporting structure or an anchor in which your epithelial cells can grow, or rather anchor to and grow around. And likewise, your endothelial cells, they also form a matrix for those endothelial cells. Remember, they're the ones that line your blood vessels. So we've classified our epithelium based on shape. We can also classify them on the number of cell layers. Okay, so we can be simple, which by its very nature has just got a single layer, you can see that, and stratified, where we have several layers. But as well as that, you have what is called pseudo-stratified. And really what that is, it's a single layer of cells, but it looks like it's got lots of layers. So in other words, it's single layered but appears to be stratified. It appears to have several layers to it. So it just looks like a multiple layered cell epithelial lining. You also have your transitional, and these may appear cuboidal or columnar when they're not stretched, and then they go on to form this squamous shape when they are stretched. So what I do in all of my recorded sessions is I really want you to have little points where you press pause on the recorder, a bit old-fashioned, but you press pause on your computer, and you just think about the questions that I'm posing to you. So what I've got a whole load of diagrams, simple little diagrams for you there, and I just want you to describe the characteristics of each epithelium layer. Okay, so for example, the first one, simple squamous, hopefully you would have said a single layer that appears flattened. Okay, so now press pause and do all the rest of them, and then I'll carry on. Okay, so your simple cuboidal. As we've already said, it's a single layer and it's cuboidal or square in shape in 3D. Your simple columnar, again, we've done this. A single layer looks like a column. Your stratified squamous. So now we're looking at this, you're thinking multiple layers, hence the stratified, squamous, therefore flattened. You've got stratified cuboidal. Again, multiple layers, cuboidal in shape. Pseudo-stratified columnar. Remember what pseudo-stratified means? It's a single layer of columnar cells, but it looks like there's more than one layer. And finally, your transitional. These are squamous when stretched and cuboidal or columnar when they aren't stretched. And basically what these do is they allow cells, these transitional cells allow stretching of things like your ureter and your bladder as they fill, and then the urine, as they fill with urine, and then they recoil as they empty. And so that's why they're nice and stretchy. They're transitional. They change that shape. So another little thing you need to be thinking about is whether they're ciliated or non-ciliated. And cilia are just little projections. As you can see down here, you can see the little projections coming off the top of these little finger-like projections, little hair-like things. And there you can see them on this right-hand diagram as well, these little hair-like projections. An example would be in your respiratory system. So for example, most of your respiratory epithelium consists of ciliated pseudo-stratified columnar epithelia with your goblet cells. So what that means, remember, cilia, you've got these little hair-like projections. Pseudo-stratified, looks like it's more than one layer, but in fact is just one layer. They're columnar, so they're tall and long, so to speak. And they have goblet cells. So we're in a respiratory system in this case, and your goblet cells, there they are in this diagram, looking at your respiratory system, and these produce mucus. And if you're thinking about what the mucus is doing, of course, it's grabbing any particulate matter, so dust and things like that. And so you've got these cilia, in the case of the epithelia, that actually can move. Okay, so what's happening is mucus production that helps to accumulate and grab hold of the dirt, the dust, and any debris that might be around. So it sticks to this mucus layer. And then the cilia, these little projections, they move in unison towards the pharynx. And so in other words, they're taking it, if they're in, if they're in your bronchi, for example, they're moving upwards towards your mouth, where you then go and swallow it. So you're removing the debris that way. This is called your mucociliary escalator, and it is always towards the mouth. That's the important thing. So if it's in your bronchi, it's coming upwards away from your lungs towards your mouth where you swallow it, and in your nose, it's going downwards towards your mouth where you swallow it. So it's called the escalator, and it's always towards the mouth. There are other examples of ciliated simple columnar. So, for example, they're found in your fallopian tubes, where the ciliary action helps facilitate the transport of the egg from the ovary towards the uterus. So we're still going on these different characterizations of epithelia. So it may be what's called keratinized or non-keratinized. And your keratinized epithelial cells, they're dead cells, they're very flat, they don't have a nucleus or any organelle, so they don't have a nucleus with the DNA in it, they don't have your organelle, so your ER, your Golgi, etc. And they're filled with a protein called keratin. And this keratin is what makes your skin waterproof. So, for example, the palms of your hand or the soles of your foot, for example. Now, what I'd like you to have a little think about, here's a little table. I'm going to want you to pause it when I've stopped speaking, but have a little look through it. And all I really want you to think about in each cell type, so I'll give you the first one, you've got squamous, simple epithelia. It's found, remember what squamous is, flattened. It's found in your alveoli and your blood vessel lining. Extra notes for you, just additional little points. You have they're involved in their surface specializations. So your microvilli in the GI tract, for example, to increase surface area and aid in absorption. And the cilia that we've already talked about in the respiratory system. So really in each case, so your cell type and then your locations and any additional notes, just want you to think, is diffusion, absorption and secretion across those epithelial layers, is really that going to be fast or slow? That's what I want you to think about, just fast and slow, or very fast and slow. And does it give you any mechanical abrasion protection? So does it protect against knocks and bumps that's going on within that tissue itself? Okay, so really all I want is, yes, good abrasion protection, or no, not really, not particularly good. So again, press pause, have a look at each of these, okay, and have a little think about, is diffusion, absorption, and secretion fast or slow, and do we get good protection against abrasion, yes or no? And again, you might not know this, but you can start to use some common sense by the very location of it and what's going on in that system. So press pause. Okay, so the first one, squamous simple epithelia. So that's in our alveoli and our blood vessels. So do we have faster fusion, absorption and secretion? Well, we're thinking about our blood vessel linings. We're thinking about our alveoli. We're thinking alveoli are in the lungs, of course, and they're involved in oxygen and CO2 exchange. So, of course, diffusion is very fast. That oxygen has to get from our alveoli into our blood very fast, and the CO2 out again very fast. So they're very thin. Do you think they have good mechanical abrasion protection? No, very little abrasion protection. They're very thin and delicate. We now have our cuboidal simple epithelia. So we see these in our kidney collecting ducts. That's the end part of our kidneys, if you like. That's where you get the fine tuning of reabsorbing water and other nutrients, or electrolytes rather, that you may need, or secretion of electrolytes if we don't need them, or secretion of water if we don't need them. Also in your salivary ducts. So they have excretory, secretory and absorptive functions. So what about diffusion across our kidneys, for example? Well, yes, that has to be pretty fast. Okay, so they're still quite thin, not as thin as the squamous, which have been flattened. So they're a bit bigger. So they're cuboidal. So they're still fast, but not as fast. Do they have any protection against abrasion? No, not really. Not very much at all. Now we'll move on to our columnar simple epithelia. So again, how fast are they diffusion? Well, again, they're relatively fast because they're found in the GI tract where we're absorbing lots of nutrients. So our absorptive surface in our small intestine, that's what SI stands for, and our LI, our large intestine and gallbladder. So what about mechanical abrasion? Again, not a lot of mechanical abrasion protection. They're single layers as a general rule, and single layers don't have a lot of mechanical protection because it's just a single layer. So now we have stratified epithelia, which you might find in your oral cavity where lots of food is coming in, your pharynx and your esophagus where you're swallowing lots of stuff, your anal canal and orifice where lots of food is being passed out, and in females, vagina, cervix, and in all of us, obviously, skin. So in those areas, do you think we're going to get fast absorption? Do we absorb much in our oral cavity or in our esophagus or our anal canal or our vagina or cervix or skin? Are we absorbing and secreting very much as a general rule? Well, no, we're not. So therefore, that speed is relatively low. Do we need to protect against mechanical abrasion, all that food coming in? All that food being swallowed down our esophagus, all the waste being passed out through the anal canal, the skin, all that abrasion that's going on there as you're wearing clothes, whacking your skin against stuff, for example. Do we get mechanical abrasion? Yes, we do. We get good mechanical abrasion. We've got lots of layers of cells there, hence it's stratified. So we're getting that good protection. So your stratified epithelia is adapted to withstand the constant abrasion to which the body surface is being exposed. So during most of my sessions, I also give you some review questions that I want you to think about at the end of a little section, if you like. Sometimes it'll be simple recalls. Sometimes you'll have to just think a little bit more. So again, what I want you to do is just press pause, have a look at the review questions, and say which ones are true and false. So there might be none of them may be correct. 1 might, 2 might, 3 might, 4 might. Okay, so you're not just looking for one correct answer. Any number of answers may be correct or any number may be wrong. Okay, so take a look. Okay, so which of the following regarding the epithelia are correct? Squamous epithelia are thin, which allows for rapid diffusion of molecules. Yes, that's correct. That was in that last table that we did. They're present in the alveoli, for example, for rapid oxygen and CO2 transport. The majority of the respiratory tract contains stratified columnar epithelial with cilia, which help remove particles bound to mucus. That is incorrect. Now, they are columnar, they are epithelia, they do have cilia, which do help to remove particles bound to mucus, but they are not stratified. There are not multiple layers. There is a single layer that looks like it's more than one, so they are pseudo-stratified columnar epithelia. The next one, exocrine glands, such as the salivary glands, are connected to surface epithelium by ducts. So that's correct. So your glands are formed by invaginations of your epithelial surfaces, which are formed during embryological development by the proliferation of your epithelium in the underlying tissue. So they're all part of that epithelial tissue, and that extension, if you like, forms the ducts and then into the glands. Endocrine glands secrete hormones into the blood and therefore have no connection to the epithelial surface from which they developed. And that is correct. They don't have that surface connection to epithelial cells from which they developed. An example is, of course, the thyroid gland. Your thyroid gland secretes thyroxine, amongst other things. Thyroxine is also known as thyroid hormone, and that helps to regulate your metabolic rate. Okay, so we're still on the epithelia, of course, because this entire session is on the epithelia. And when you're looking at it, you need to have a continuous cohesive layer within your epithelia. So here we've got it. So we've got 1 cell, 2 cells, 3 cells, 4 cells, all forming this nice cohesive layer. But of course, they're all communicating with each other, but things need to cross them as well. So what you get is you have a variety of different cell junctions formed by the adjacent surfaces of epithelial cells to help form this cohesive layer. And there are lots of different types of cell junction. And what this is actually doing is enabling the epithelia to form barriers to the movement of water, solutes and cells, for example, from one body compartment to another, or to enable movement between adjacent cells as is required. So first of all, if we look at the first example here, so there you go, you've got a tight junction, and that forms a watertight seal between two adjacent cells. And so the purpose, or the overall purpose of tight junctions, is to keep liquid from escaping between cells. It's a tight junction. And this allows a layer of cells, for instance those lining an organ, to act as an impermeable barrier. For example, the tight junctions between the epithelial cells lining your bladder prevent urine from leaking out into the extracellular space. You don't want your urine leaking out of your bladder. Desmosomes, there you've got another one on the left hand side. So these pin adjacent epithelial cells together to ensure that stretchy tissues remain in an unbroken sheet. So, for example, what they're working on is tissues such as your skin and your cardiac muscle, and it helps to allow them to remain connected in an unbroken shape as they stretch away. Then you've got your gap junction, so that is shown there, and that creates a channel between neighbouring cells. So this cell is now talking to this cell by the flow of molecules between the two of them along this gap junction. So it's a communication junction, and it allows for the transport of ions, water and other soluble and other substances. And it allows or permits the coordinated movements, for example, of muscles leading to childbirth within the uterus, or the talk between the cardiac myocytes, so that's your cardiomyocytes, your cardiac cells, your heart cells, leading to coordinated and simultaneous heart myocyte contraction. So simultaneous heart contraction of all of the cells within it, within any one region of it. So we've already talked about the cilia actually when we talked about the respiratory system. So these are actively motile structures that are easily seen, just so you know how big they are, they're easily seen by a light microscope. And you then have those cilia, but you also have microvilli. So microvilli are shorter projections of the plasma membrane, and for example on the absorptive surfaces of the small intestine, so to increase surface area, allow you to absorb nutrients into the small intestine, whilst your stereocilia, which is shown in this little diagram up here, are extremely long. There they are, stereocilia, microvilli, found in small numbers but aren't motile. They move, in the case of the stereocilia in the inner ear, due to fluid movement, and they're to do with balance, for example. So when you're spinning your head round and round and round, you have these little stereocilia. They either move one way when you spin one way, or if you spin the other way, they move the other way. And that basically is all being processed through your nervous system, and that then tells you, your body, which way you are actually spinning. For example, if your eyes were shut, okay, and you can't see which way you're spinning, that balance system would allow you to know which way you're going. So those are what your stereocilia are doing. So really, they're sensory hairs. And as I mentioned, underneath all of these structures, we have the basement membrane. And the basement membrane is providing your structural support between the epithelia and your endothelia and the underlying supporting tissues. So there you've got your epidermis, your skin, and there's your basement membrane layer. Okay, and you can see there hairs coming out, and there are nerves all around, etc. And so your nutrients for the epithelial cells must diffuse across this this non-cellular basement membrane. There are no cells in there. Okay, and that's because your nerve fibers, they can penetrate the basement membrane, but your small blood vessels don't usually enter the epithelia.